Yes, hello. It's going to be difficult for me to follow this presentation because I'm actually looking at sort of similar images as Dario has just presented, but I'll do my best. It's certainly very nice to be part of this meeting. I had, to be honest, never heard of cytodata, but it seems like a very interesting community to become part of. And I'm very much looking forward to the rest of the, coming 3 days, particularly the hackathon, actually. I would like to join with that. So my name is Hanneke. I'm part of the Babraham Institute imaging facility, where I work as an image analyst and our high content imaging specialist. Now you may wonder why I have a picture of spoons. Well, I chose this picture because it sort of appears to me to be kind of an illustration of my job as an image analyst. People bring me their microscopy images, and it's up to me to find from a huge selection of very nice tools, the right one to analyze their data. And it's not always that, why is it not going? It's not always that easy, because within our imaging facility, I get a huge variety of images. This is because although we are just predominantly a light microscopy facility, we do have a number of different imaging modalities, so confocal and high content, multiphoton, etc. But mainly the reason for the variety of images is the variety of fields that our facility users are interested in. So we have people interested in apoptosis and cancer and various things. So our users are mainly from the Babraham Institute, 3 departments. So there is an epigenetics, an immunology, and a signaling department. But we also have various users from companies on site, and they could be focusing their research on various other topics. So on the screen right now, you can see an example of, many examples of the different kinds of images that we would get. And of course, the talk, being only 10 minutes, would be way too short to go into all of them. So I've just selected this one over here to concentrate on. And the main thing would be, the main message that I would like to give regarding this image is that it's a germinal center. Germinal centers are specialized sites in immune tissues where B and T cells, but other immune cells as well, work together to generate an immune response. And you can already see from the image that the organization of these sites is very complex. So there are different cell types that are in very specialized locations. So this image was a 7-channel confocal image captured on our Leica Stellaris confocal microscope by Sigrid Frabado in the Lintemann lab. And so the Lintemann lab looks at these germinal centers. And you can more clearly see the complexity of this site by showing the image channels, the 7 image channels separately. And what you notice, for instance, is that cells can locate to different compartments. They can have a very specialized localization pattern. And it's sometimes even the case that, for instance, when you look at naive B cells, they are well, predominantly excluded from the germinal center, whereas actively proliferating B cells are actually localized in the germinal center. So my job as an image analyst would be to determine, for instance, how big these centers are, because with age, for instance, the size of germinal centers decreases. It would also be an interesting question to see how many cells of which cell types are localized in the germinal center, and how are these cells interacting with each other. Yeah, kind of a simplified version of what Daria was just talking to you about. Now, to tackle this question, a good place to start, obviously, would be to first of all look at the center itself. Create a segmentation mask of the germinal center, and then one by one look at the different cell types and generate segmentation masks for these different cell types, so that you can pinpoint where exactly each individual cell is located. Now, what would have made that job a lot easier would have been to have some kind of nuclear counterstain, which unfortunately is not present in this image. You may notice the sort of nuclear shapes in the blue channel. So this is the BCL6 marker that stains for proliferating B cells. But all the other markers are cell surface markers. So it's a little bit trickier to actually find those cells without actually anything being inside the nucleus. Now, to do this, there's of course various options of analysis software, and I'm not going to show you ways to do it with all of these, but potentially you could use Fiji, you could use Cell Profiler, you could use QPath, or you could use CellPose, or you could use whatever software comes with your microscope itself. For the purpose of this talk, I've used Fiji, and particularly I've used the StarDist plugin. If you are not aware of this, it's a very nice Fiji implemented trained neural network for detection of nuclei in 2D mostly, but also 3D. It was developed by Uwe Schmidt and Martin Weigert. Link over here. So questions that you then may start answering would be how big are the germinal centers, and how many proliferating B cells are inside. So it would look a bit like this, and it would look a bit like this. You could find out the number of T cells and the number of T cells that actually are activated, and the number of Tregs. And you can then again find out how many of these specific cells or cell types or cell subtypes are located within the germinal center. And then, of course, once you know the location of these cells, you can, just by simple trigonometry, calculate the distance between neighbors and the identity of the neighbors. And you could do this, of course, different ways. So you can calculate exact distances, or you can say, okay, if I go in a radius around each cell, how many cells of other cell types are found within this radius. So for instance, the B cell in the center of this image has been surrounded by 6 T cells, and in red and in cyan. So 2 of which are regulatory T cells, and it also has within the circle 1 B cell, while it has a number of other neighbors on the edge of the neighborhood. Now, this can, of course, be used not only for tissue sections of germinal centers. You can use it for other tissues as well, also for multiple colors. We are going to, well, we have recently secured funding to acquire an ultra high-plex immunocytochemistry platform. So potentially we could get images in many more colors than just the 7 that we can acquire on the Leica microscope. And but thinking also more wildly, perhaps you could use a similar kind of analysis pipeline to look at organization within nuclei, for instance. So chromatin folding and how histones are organized around this. And this, I guess, would be particularly interesting for our facility users within our epigenetics department. This was my talk. It was maybe quite short, but thank you for listening. I look forward to the rest of the conference.